Hello, and welcome to the second part of our screencast on the cell cycle. This part will be on control of the cell cycle. The cell cycle control system. The cool thing about cells is that they do what they need to do only when they need to do that. I always think of them as that they are very smart. Really, what it is evolution and natural selection that cells are efficient. We will at some point talk about how genes are regulated, so certain genes are turned on when that stuff needs to happen. Related to that is how the cells are going to know when to divide. There are three main checkpoints. And so if the cell gets the green light to go through that checkpoint, well, then it will continue through the cell cycle. If there is something wrong in the cell, like there's a mutation in the DNA, there are too many cells in an area, or there's not enough nutrients, or the cell is just where it needs to be, it could enter the G0 phase. Example of cells that are in G0 in humans would be nerve cells and muscle cells. Also liver cells, but liver cells, when damaged, can go back into the cell cycle. As an example, the G1 checkpoint takes place at the end of G1. So once the cell has gone through G1, it has gotten larger after it just divided in half. If there is enough nutrients around, everything else is functioning correctly in the cell, then the cell will go into the S phase and replicate its DNA. Replicating DNA is pretty important. If the DNA is not replicated and replicated properly, so no mutations, then the cell cannot go through the cell cycle. And so one of the other checkpoints would be to make sure the DNA is copied through the S phase. Okay, the internal control of the cell cycle. We're going to look at the proteins that are present in varying concentrations in the cell cycle that will actually determine whether the cell goes through a checkpoint. Because remember, the cell is not just like a car going through a racetrack and it has a green light and keeps on going. Instead, there is a system of protein controls that will signal when the cell it needs to do something. Cyclins and cyclin-dependent kinases are the primary things that are going to control the cell cycle. Cyclins are proteins, and like the name implies, they're going to cycle in concentration through the cell cycle. If you look at this graphic at the bottom, the pink line shows you the concentration of cyclin. So as the concentration rises, so as a cell is synthesizing more cyclin, that's going to trigger mitosis. At the end of mitosis, you notice how it drops off sharply? Well, mitosis is also ending. So that must be what causes the mitosis to end. And then as we go through interphase, cyclin concentration raises again, go through mitosis, and so on. Okay, then CDKs, cyclin-dependent kinases, are always present in the cell, but they're only active if they are attached to a cyclin. This is a convenient thing for the cell, so it doesn't have to make new CDKs. They're always there. All the cell does to regulate is to make the cyclins. So if there's no cyclin, if you look over here on the left, there is a CDK, but it's not active. Then once a cyclin binds to the CDK, it becomes active. It will then phosphorylate whatever targets it has. The example here is showing how it's going to start the S phase. Phosphorylating is really important that we'll see several times. Essentially what that means is that you're activating something. We're adding a phosphate group. Of course, that would come from ATP or another form of something, GTP, AMP, other things. But the phosphorylation will actually activate something, and that's what will cause it to go and do what it needs to do. Okay, so an example of a CDK is an MPF. MPF You can actually find that it stands for different things, but let's just go with mitosis promoting factor because it's going to promote mitosis. Okay, so here's a similar graphic to what we just had, but if we look at MPF activity, um, it's only going to be active whenever cyclin is active. So if I were to add in a line, I'm sorry about that. In CDKs are always going to be present in the cell. So that's going to be a straight line across. But the cyclins are what? Cyclin concentration. So as the cyclin bonds to the CDK, this is going to cause MPF to become active, which means it's going to cause mitosis. Okay, so details for actually what the MPF does. So the cell makes cyclin during the S phase. As that cyclin is made, it combines with the CDK to make the MPF. Once there is enough MPF, remember there's not just one of these floating around the cell, this is on the scale of trillions. Once there's enough active MPF, the cell can begin mitosis. But it's not just some sort of green light saying go ahead. MPF actually does very specific functions in the cell, such as breaking apart the nucleus and building the mitotic spindle. And then the cyclin begins to degrade during anaphase. In other words, the mitotic spindle will begin to degrade and the nucleus will begin to reform because there's no MPF to make all of this stuff happen. So, for another application of this, if MPF did not become inactive, in other words, cyclins stayed where they were, mitosis would come to a stop. This means that once we got to metaphase, nothing else would happen. Okay, so short summary, no cyclin, no MPF, no mitosis. CDK is always present, just not always active. Cyclin cycles in concentration. Okay, now for external control. This is a little bit more straightforward. Basically, this is going to be where you have proteins and other possible environmental signals generated by other cells that will signal to another cell if it needs to divide or not. In our lesson on cell communication, we will get all into what this stuff in the middle, what's going on here. What we're looking at, just the basic idea, is that there's going to be some sort of signal from the outside of the cell that's going to trigger a response inside the cell. That response is going to be either to start or halt cell division. One example would be platelet-derived growth factor, PDGF. Biologists love to name things and then make abbreviations. These are growth factors. If you have ever had a cut and it scabbed over, that was a result of platelets accumulating at the open wound. We have proteins called fibrins that are going to build there, and this is what forms your scab. The platelets release this PDGF to stimulate cell division. I'm sorry, there was a bell. Again, platelets will release the PDGF to stimulate cell division in the damaged tissue. So the platelets get here, they release this growth factor, and that growth factor is what makes your skin grow back. By the way, this is an example of positive feedback. So as the platelets get here, they cause more platelets to arise and more cell division to occur. Okay, two other examples, density-dependent inhibition and anchorage dependence, are actually what they sound like. So anchorage dependence, most cells have to be attached to some sort of substrate in order to divide. Density dependent, this means that once we have too many cells in an area, they're going to stop dividing. A tumor can result if one of these things do not happen. A tumor could then become cancerous or not. And then cancer is obviously not that common, even though we all hear about it a lot. But the basic idea of what causes cancer is just a disruption in the cell cycle control. And so what causes this would be a mutation in one of the genes that controls the cell cycle. Now, you have mutations all of the time. This is not an uncommon thing. Mutations don't always lead to cancer. Your cell has lots of different self check mechanisms to take care of mutations. If there is a mutation in DNA replication and the cell is not able to repair it, these mutated cells should commit cell suicide, the apoptosis. And this chart right here is showing you some of the characteristics of cancer cells. It's not just an overgrowth of cells. The cells that result, they don't have any more error checking mechanisms, so mutations are going to accumulate. Cancer cells actually physically look different. They can release toxins, they can interfere with your other organs by driving blood vessels and even just impeding their movement because if the tumor grows large. Okay, so proto-oncogenes. This is one of the genes that controls the cell cycle. A proto-oncogene is something that everybody has. Proto means before, onco refers to cancer, like an oncologist. If there's a mutation in the proto-oncogene, we then call it an oncogene. In this case, the gene is always on. This means that the gene is telling the cell to always divide. So proto-oncogene is the normal one. This is the one that will make cyclins and CDKs. It turns on and turns off. But if it mutates, well, now the gene is always on. This causes an overgrowth of cells, and that can develop into cancer. Another example of a gene that controls a cell cycle would be the tumor suppressor genes. These are going to inhibit cell division. So these are like the brakes. If there is mutation in one of these genes, then the gene is always off, which means you have no brakes on your car, so the car continues to go or the cell continues to divide, and this again can lead to cancer. A side note with both of these are a class of genes. There's lots of different tumor suppressor genes and proto-oncogenes. There's lots of possible mutations that can occur in each of these, which results in any type of cancer being almost unique between affecting different organs. For example, breast cancer versus leukemia versus a brain tumor. But even beyond that, everybody's mutation that's causing that cancer is different, which makes cancer kind of difficult to treat. The paramecium parlor, the cell checkpoint, all violators must fix their mistakes or face apoptosis. In other words, if the cell doesn't do what it's supposed to do, well, the cell's going to die. And last, biology is the only science where multiplication and division mean the same thing.